main challenges from microbiome studies are to understand the originality of the human microbiome. In fact, we do as if humans were very similar to all animals and so on. And for example, regulation systems are imposing models which are completely foreign to what we are. We must understand that we evolved in a way where the ancestors of Homo sapiens tamed fire. This means that we all eat cooked food, and this we did for millions of years. And no animal has done that at all. So this means that there is a very specific originality of our microbiome, which is that we eat cooked food, and cooked food means aged food. It's very similar. The chemistry of aging, chemistry of cooking is the same, more or less. And in fact, so this means that we have adapted to aging in a way which is quite different from other animals like our primate cousins. And this means, for example, that we live much longer than expected. Contrary to what many people think, our microbiome does not provide us with the proper metabolites that we need. In fact, we get them from microbiomes, but not ours. It is from the fact that we eat animals, for example, which is very important for two metabolites, vitamin B12, for example, and also taurine, for example. In fact, in the end, what we often discover, at least in the domain of metabolism, is that the microbes which are important are the microbes which die. They die, lyse, and at this point, their internal content is used by our cells. So this means that the different categories of microbes which are important are usually not those that people think about. And also it tells us something very interesting. You know that there are viruses which kill microbes. They are named, in terms of, in the case of bacteria, they are named bacteriophages. So this means that the role of bacteriophages is very, very important because they will lyse the microbe, expose their content, and make their content available to the animals, plants, and so on. There's also another feature which is not understood, is the fact that because these communities are close together, they exchange the genes all the time. So this means that if you study a species, maybe this doesn't mean anything, because the genes of this some genes of this species, which would be important for producing something, may be transmitted horizontally to other species. So we should study microbiota not in a species-orientated or strain-orientated, but in a gene-orientated way.